Yeah, just on.
I know you're good to go.
Okay, so hi everyone, welcome to the CAMBAM QLS joint seminar. For today's speaker, we have Dr. David Sept from University of Michigan in the Department of Biomedical Engineering. I discovered today that he's actually Canadian, which is kind of awesome.
That too.
Yeah. So David, before actually he was at the University of Michigan, he was University of Washington, and prior to that he did
Close, Washington University, but yeah,
Washington University. Sorry, the other way.
in St. Louis.
Yes, yeah, and he did his postdoctoral fellowship at the University of St.
Yeah, UCSD and Salk.
So yeah, and Salk as well. And prior to that, he did his PhD at the University of Alberta. So I'm looking forward to hearing your talk, and the microphone is yours.
Great, thanks very much. Question is this, can I don't move that, so.
You can click OK.
Yeah, the thing is, I don't know if I can get over there. Let this escape up for a second. No. All right, if that's the only technical challenge we have today, that would be great. So thanks very much, Omar, for the introduction. I'm really glad to be here. This is a pretty diverse group of people, and I want to treat this as a pretty informal presentation. This is kind of a making of a lot of different talks that I've given, trying to incorporate some new stuff with a slightly different theme. So there's a good chance there'll be a little bit of a kind of a non sequitur at certain points. Please do not hesitate to ask questions as we go along. I'm certainly open to having that, and I think if you don't understand what's going on, if you get that clear, understanding, you'll probably enjoy the rest of the talk. So start off with a little bit of background. So microtubules are key components of every cell. So cells have what's called the cytoskeleton, as you might imagine. The skeleton is actually what gives structure to the cell, but it's also a very dynamic construct which can reorganize and help cells perform a lot of their different functions. There's three primary filaments that go into the cytoskeleton. There are actin filaments, which are shown here in red. Actually, there's intermediate filaments, which we can't see. Sorry about that, Morgan. And then there are microtubules here shown in green along the cell nucleus. And these three sets of filaments actually work in consort in a lot of different functions. One of the main things that microtubules do is they provide a mechanism for transport. So there's motor proteins that actually walk along these filaments. There's kinesin, which, for example, would carry things in the direction, out towards from the microtubule organizing center out towards the membrane of the cell. There's dynein, which would move in the opposite direction. They're also very important in axons. Microtubules, there's dense arrays of axons that enable for trafficking kind of up and down the axon as well for materials. And then there's actually a whole series of microtubule associated proteins which would bind and modify the protein, which would cross-link things. Maybe you've heard of proteins like tau, which has been implicated in Alzheimer's disease and other things such as that. Right. So this is another standard textbook view of what microtubules do in terms of motility. So if you were to take a cross section of a flagella or a cilia, something, you would see these kind of patterns of microtubules that go up. And these provide either kind of the rotary motion or the beating motion that you would see that cells use to propel themselves around. This is not a cell biology textbook view, but this is a painting by Frida Kahlo called Moses. And one of the things I like quite a bit about it is here just above, so you see Karl Marx and Nefertiti, but then they have a very nice picture of a dividing cell up there. And microtubules are also involved in mitosis. When cells divide, the microtubules come out, attach onto the chromosome pairs, basically separate them and pull them apart. So you see here that they're being kind of pulled apart in this phase, and then here, then this is kind of, late stages of cell division. And they basically separate the chromosomes, divide off the two cells, and then you've kind of completed your cell division. Okay, so the building blocks of microtubules are tubulin, right? These tubulin dimers. This is a heterodimer of alpha and beta tubulin. So alpha here is in the bottom, beta tubulin is on the top. Each one of these binds a copy of GTP, okay, is the nucleotide. And as you might imagine, just because of the proximity, you see that that nucleotide in the bottom is kind of buried within the dimer, but the top nucleotide in beta, it actually can be hydrolyzed, which is important for its function, and it can also then be exchanged in solution to get a fresh copy of GTP. Okay. So if you take these dimers, then you can assemble them into the microtubule. So this is the canonical view, if you will. So each of these dimers is about 8 nanometers in size, and the tube itself is about 25 nanometers across. Okay, the dimers arrange themselves in a head-to-tail fashion. So you see that each dimer has an alpha on the bottom and a beta on the top. So they have they're polarized in these arrays. They attach together, and there's 13 of them in a typical microtubule that would come together to form the tube. Interestingly, they're not kind of stacked in rings, but it actually follows a helix. So if I start here on a beta subunit, I run through beta, 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 all the way around. I come up here, beta, beta, and then I hit an alpha. So this is what's called a 3-star helix because I end up 3 monomers above where I started before, okay? And we're going to be very interested in kind of looking at longitudinal interactions as well as lateral interactions between the protofilaments. One of the unique features of this is because there's 13 protofilaments and because there's this 3-star helix, we have a discontinuity here, which is called the seam as well. So instead of having beta, 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 alpha, alpha, example, now we have alpha going to beta and beta going to alpha. This discontinuity will not feature very much in today's talk, but it's an interesting component still. So over the last almost 20 years, we've really learned a lot about what makes tubulin special. And there's this whole idea that's come out, which is called the Tubulin Code, which followed quickly on the movie of the same name you maybe saw from 2006. This is an idea where there's some sense that tubulin is marked or indicated or bears some type of code that actually relates to its function within the microtubule. Okay? And this tubulin code is a combination of 2 different things. One is different tubulin isotypes, okay, of alpha and beta. And the second is this range of post-translational modifications. Okay, and we'll talk a bit about both of those pieces now. Okay, so tubulin isotypes in humans, we have 8 to 9 isotypes of both alpha and beta. We know that some of them play specific roles. For example, in muscle cells, we see a lot of alpha 8. In neurons, we get a lot of the beta 2 family as well as beta 3. Ciliated cells, again, have a different set, and platelets tend to feature beta 1. But what's kind of interesting is when you look at these across species, which that phylogenetic tree on the right shows, you see that we see a lot of conservation, high degree of sequence identity between all of these isotypes. Even in kind of some of these lower organisms, so yeast is maybe a little bit further off, but things like fly, zebrafish, where they actually have 11 isotypes in this case, okay, we see a high degree of sequence conservation. This is just for alpha tubulin, but you can imagine basically the same set of isotypes for beta tubulin. And because of the combinations now, when I take an alpha and beta and put them together to make a heterodimer, you can see the vast diversity that we get, right, of probably on the order of 70 to 80 different type of isotypes. There could be rules that prevent one isotype from binding to another. I don't think we have any sense of what those rules are yet. This is the first part of the code. The second part of the code is post-translational modification. You're probably familiar about this. Kinases and phosphatases that kind of modify proteins after they've been translated. Tubulin has a broad range of PTMs. We're still discovering some of them. One of the first ones that was discovered was actually acetylation of a lysine in alpha tubulin. It was discovered about 30, 40 years ago now, I guess, in kind of the mid-80s. But some of the more interesting ones are all happen on these C-terminal tails. Okay, so these tails that come off of tubulin are very rich in glutamates, you'll see in a little bit, but then they typically end in alpha in an EEY motif. So first of all, there's enzymes that will come on and basically cleave off this terminal tyrosine, what's called G-tyrosination. Then we can form a delta 2 by having this terminal glutamate removed, and even delta 3 by having that next one removed. There's also these extensions that can come off, right? If I've got a glutamate there, it has a carboxy side chain, which looks exactly like a C-terminus of a peptide. There's enzymes which will then come in and polymerize a new, basically, peptide chain off of that. So I can have polyglycylation, which forms, or polyglutamylation or polyglycylation. And that can happen on both alpha and beta. The most common level of glutamylation that you see is typically having one glutamate on there. It's actually, and then, but you can even see chains of up to, I think some in the literature I've seen some, I don't know, up to 17, 20 or something like that. We're gaining little bits of insight into how this functions in the cell, but I still don't think we see that much. Mass spec is kind of been a game changer in how we're learning to find these types of PTMs. It's very, very challenging in this case because the isotypes, the proteins are 95% identical in terms of sequence. There's very small regions where they differ. And that actually makes the mass spec different, hard to do because you can't tell which part came from which protein. Okay, so a little bit back now, looking at molecular structure of the microtubule. And we're going to construct a very small system, basically made of 2 neighboring protofilaments with 6 of these monomers. This is actually an old slide, as you can tell from when I thought Comic Sans was a good choice for a font. But this is a set of old simulations that we did, basically looking at the contacts and kind of the mechanics, if you will, of this system. And a couple of the key loops that we're going to be talking about are what are called the M loop and the H1S2 loops, shown here in red and yellows, respectively. This is the outside view. If you go to the inside view where these loops are, you can see them a little bit more clearly there on the right. Okay. So we're going to take this system and we're going to perform what is called molecular dynamics to kind of look at its at the dynamics conformations and its properties, if you will. So MD is a system where basically we're just integrating Newton's equation of motion, F equals ma. Okay, we know what the masses of these atoms are, and the forces come from what's called a force field. And the force field contains a series of terms. The first is that for any atoms that are connected, that are directly connected by a covalent bond, we have a bond stretching term, which defines kind of, almost think of it like a spring, how much that bond length can change. We also have for each group of 3 atoms in a covalent link, right? Then we have an angle bending term. You can imagine things like a water molecule or any one of these connections within the peptide chain. And then for all 4, then we have a torsional degree of freedom, where basically we have rotation around that central bond for all groups of 4 atoms. Then for proteins that are more than 4 atoms apart, right? They don't have these direct, what we call bonded terms, but they have non-bonded interactions through Coulomb's law, basically through the electrostatic term, and through van der Waals interactions, where we use basically like a Leonard-Jones type of potential. Okay, so we provide this force field, right? We basically add water and ions. We set the pH of the system. We define the pressure and the temperature, right? And then basically we just simulate the dynamics, and the thermal energy that it gets from the temperature that we apply basically provides dynamics to the system. Right? So these are the simulations that were done. This is done amazingly 20 years ago now. It makes me feel even older. But this was kind of a state-of-the-art simulation for the time at which we did it, right? This was 30 nanoseconds of data on what was a relatively large system at the time. We got early access to this new supercomputer, and we're able to run this stuff. And this molecular level simulation provided a lot of interest and insight, but for today's purposes, we're going to take a slightly different tack. So we're going to take the results of that MD simulation, and we're only going to look at the protein part of it, okay? And we're going to coarse grain this, where we're going to take the center of each one of those monomers and basically treat it as a sphere. And so we're going to reduce this whole system and the dynamics to the dynamics of 6 spheres within the system. And using basically thin shell mechanics, we're going to look at the bending of that system, compression along the length of the protofilaments, and then the shear between protofilaments. Okay, these very simple mechanical bones. Now, there are ways within MD where we could apply forces, do non-equilibrium MD. We were not smart enough to think of that before we ran these simulations, but now we can kind of come back to them after the end, after we have the results, okay, and basically do analysis looking at the equilibrium fluctuations that we see. So for example, for the bending mode, right, that's a 1 degree of freedom, and we know from equipartition theorem that that has kT over 2 of energy, okay? And so since we know what our temperature is and what the parameters of the lattice are, then we can measure the fluctuations in that bending angle, get the variance, and from that variance, then we can calculate the bending rigidity directly. That's shown as kappa over here. Okay. So when we do that for our system, we actually find that here's our normal microtubule versus a Taxol bound microtubule, right? It's known experimentally that Taxol softens the microtubule, that it actually decreases the persistence length. It makes it more compliant in some sense. And we see that here because we get a larger range of motion within Taxol. The compression, same thing. Taxol is more compressible in the same direction. And when we take both of those parameters using, again, some assumptions out of thin shell mechanics, we actually can recover the correct Young's modulus for the system. We get one of the things that comes out is the thickness of the wall, which is on the order of 4 nanometers. And more interesting, even though we're simulating a system that's on the level of 12 nanometers in size, right? It does give us the right persistence length, which is actually about 6 orders of magnitude higher, up in the millimeter range. Now, compression and bending moduli are fine and interesting, but one of the things that always perplexed us was that the shear modulus didn't change. And in fact, the shear modulus was really anomalously low for what you would expect in this system. The microtubule has been known to have anisotropic mechanical properties, right? There's a famous paper that came out probably 15 or so years ago talking about a length-dependent persistence length, which actually from continuum mechanics really doesn't make any sense unless you have these really anisotropic mechanics. And so we actually suspected that this shear force, where these protofilaments are sliding against each other, was responsible for this, and that it was the loops that make these contacts between them, between the protofilaments that were you saw before, H1S2 loop and the M loop. That these were the culprits, if you will. There's another feature that's been discovered more recently, and that's relates to what's called the microtubule lattice, right? So in a normal microtubule, normal is a bad word, in a 13 protofilament microtubule in the GDP state, let's say, basically the protofilaments run almost directly up the axis of the microtubule. But what's kind of interesting is that if you change the nucleotide, so it's going from GDP to GMP CPP, you actually end up with a slight helical twist of the protofilaments, okay, going in one direction. This is not to scale, but it's just to give you a feeling of what is changing. And if you put in another non-hydrolyzable nucleotide, GDB gamma S, you actually get a shift in the opposite direction. What's important with these changes is that the protofilaments themselves are really not being shifted. If you were to look at the contacts in a given protofilament, the contacts between alpha and beta and the structure of the protofilament is not changing. What's changing is that when I'm going from straight to twisted, is that I'm getting a little bit of shear within this system. Okay, and so this is this. You can see this with these nucleotides. You can also see it when you saturate these with drugs such as Taxol. When you saturate them with motor proteins or other MAPs, that you see these changes in the lattice. So we've looked at this H1S2 loop in the past. This is actually work that was done with Gary sitting in the front row here. So for years we've worked on vertebrate. So this is the sequence of the vertebrate H1S2 loop. But then with Gary, we did work on fly, and we've done work on worm, and then with another collaborator, we've done work on yeast. And if we look at these structures, we see that there are a handful of sequence differences throughout them. You see kind of a region in here where I see variation, and then a second region up in here. And when we look at them structurally, as shown there on the right, we see that between the helix 1 and strand 2, so this is the H1S2 loop, depending on the sequence that we have here, we get different structures, right? In yeast, we tend to see an alpha helix that's formed at the top. In worm, we get these kind of beta sheets that get to be formed here and here, okay? And so we see this structural diversity. So our question was, well, what would happen, right, if we looked at these lateral contacts, at these loop interactions between the protofilaments, how would that depend on these different tubulin isotypes? Okay, so we start off, here's the series of H1S2 loops for beta subunits. Interestingly, alpha shows less diversity than beta in this region, so we decided to at least start with beta and see what we would get. Okay, and we're going to start with a couple of the key ones, namely tub B1, tub B3, and tub B4B, actually in this case. Okay, we're going to take this, put these in dimers, and we're going to basically look at the loop contacts between, at these protofilament contacts and see what we see in terms of structures. Okay. People with me so far? Everyone's nodding, but I think that's true, but okay. We'll keep going anyway. All right, so starting off with tub B1, right? So the H1S2 loop is shown here in green, right? Now this is a static conformation. As you're going to see, you can't be biased by what you see in a structure like this, because this is one conformation of the peptide with one set of secondary structures, which as you're going to see is going to change. But we see a couple different salt bridges formed. One here that forms between H1S2 loop and the body of beta, one that forms here within the H1S2 loop itself, and one that forms up here between the H1S2 loop and the M loop on the other side. If we just count the number of salt bridges that we see in a given snapshot, and we've done a lot of sampling on this, so these are pretty good numbers. You see about 10% of the time we see one, almost 27, 28% of the time we see two. 3 is the peak, and then it kind of falls off. So the mean ends up being around 2.8 salt bridges within a given conformation. Now, if we take that H1S2 loop, take the C alpha positions, so we're looking at just the backbone really of the peptide, right? And we do principal component analysis on the coordinates. This is what we get when we plot, kind of look at the first two primary modes, right? So PC1 and PC2, the largest components of the variation. So if we do contour analysis, we can see hints of some clustering in there. And if we actually apply the clusters themselves, colors are not too great on here right now, but you can see that we see at least 4 different groups that come through here. Okay. If we look at those 4 groups, we see a slight difference in secondary structure. So overall, again, this is the secondary structure, average secondary structure of the peptide. We see sheets here. This is the N-terminus, some small strands here, sorry, a small helix, strands again, a larger potential for helix in the middle, etc., as you go along. Okay. But if you look at these 4 groups, okay, then we see that this group on the end here basically has strands and no helix. This second group, the green one there and the second one there, now we've got strong helix in the middle and strong strands. As we move into the blue, the helix is similar, but now we're losing the strands, and finally moving into this last group that's lost all secondary structure completely. Okay. And each of these then, this causes rearrangements in the protein. The secondary structure is obviously changing, and it also changes in these salt bridges that we saw before in the previous slide. Moving on to 2 of these other isotypes, right? So here we saw these 4 states, right? When we move into tub 4b and tub b3, each of these seems to only exhibit 2 different states. And instead of seeing this variation in structure at this N-terminal part, it actually is all variation in these helices kind of within the middle of the peptide, right? So we're seeing kind of different structures. Interestingly, again, we saw 2.8 salt bridges on average for tub B1. For tub B3, most often it forms 1. It almost has this kind of exponential distribution. It falls off very quickly and forms very few salt bridges. Tub B4, again, like tub B3, formed 2 states, but it tends to have more salt bridges. So you're seeing kind of a set of diversity where we're trading off structure, which is affecting kind of the number of states that we can occupy, and that's affecting then the number of salt bridges that we see.
Are there functional differences between all of these?
That's almost assuredly, yes. We don't really know what they are. One of the challenges is that when people do experiments, brain is the typical source for tubulin because it's a high amount of microtubules and all these axons. You purify this tubulin and you're getting a mix of all of these different isotypes all at the same time. To express the amount of tubulin that you need to be able to test some of these is to do biochemical experiments is still a bit of a challenge. People are doing it, but not systematically, I wouldn't say that. Would you agree with that, Gary, Suzanne? Yeah.
And it's modified. It says tag.
Yeah, that's the other. Yeah, brain is also a mess because of all the PTMs, but so yeah. So what's easy is not always useful.
But are there, for example, like mutations that correspond to a disease state?
So there's a lot of what they call tubulinopathies. Anything that's significant that really disrupts the microtubules, a lot of those are embryonic lethal. But there are still certainly some that do cause disease. And some of them are amazingly subtle, right? There's one where it's normally in wild type tubulin, it's a valine, and it can lose a methyl group and become an alanine, which actually then that leads to one type of brain disorder, or it can become an isoleucine by basically adding another methyl group on, leads to a different brain disorder. So there's some subtle things that we just simply don't understand. There's 40 careers worth of work in there. All right, so just a quick summary on kind of where we've been so far looking at these lateral contacts. So the idea that the shear between the protofilaments is dependent on these loop interactions. What's interesting is when you look at the mechanics of a microtubule, it actually, as an overall unit, it actually looks it's closer to looking like 13 uncoupled protofilaments than it is like an intact tube, right? So we know these contacts are very weak. And these lateral interactions, we see dependence on the type of isotypes that we put in there, and we've only so far looked at 3 of them, so they all look different. But so far these show different structures, you know, in terms of secondary structure as well as tertiary structure, different numbers of states, and different number of salt bridges between and within the loops. Now, one of the second, oh yeah, sorry, go ahead.
So, in one of your first slides, you showed us that isotypes, some cell types, I think the isotypes are mostly distributed by cell types, if I'm correct.
So there are some isotypes that appear more predominantly in certain cell types.
But
It's,
you have some that have mixes.
almost all of
Yeah.
them will have mixes.
Were you able to look at mixes of those properties of mixes of isotypes are only all the same isotypes in the same?
So this is new stuff that's been done in the last 6 months kind of thing. So it also makes a difference whether I've got, so mixes are great. It matters now whether I've got beta 3 on the left and a beta 1 on the right, or vice versa, because I've got different loops interacting in different directions. Right? Because it's not a symmetric thing. I'll also tell you that simulations we've been doing on just isolated dimers, if I've got let's say an alpha subunit tub A1A, one of the canonical ones, and I put different betas on top of it, the dynamics of the alpha depends on which beta I put on top. So there's a huge number of combinatorics here. Yeah, please.
Jan, you said from the expression level, I think is what you're referring to, that there is some heterogeneity about what is expressed in a given cell type, which tubulin isoform. But is there any evidence that there's also like post-translational regulation that kind of maybe would make it a more homogeneous given a certain cell type? Or is all evidence pointing that it's always gonna be?
That's a really good question. So you
Everyone is listening.
yeah. So I'll give you the physicist response to that, because that's, we were talking this morning, my last biology class was in 10th grade. So there's interesting regulation that goes on at the transcription level. Certainly because if you look at mRNA levels, it looks like there's a lot of tubulin and a lot of different isotypes are there. When you look at the protein levels, that doesn't match what you see at the RNA level. So there's some level of control there. We do know a little bit about how, so the monomers are also not terribly stable on their own. So there is some control with that when they get transcribed, that they come out and form dimers readily. I don't know, are there other pieces that you would add to this? Cool. Okay. So it's there's a lot of unknowns. Is that a good enough answer? Can I get away with that? Yeah, please.
Kind of turning off that. So you said the mRNA expression levels don't match the protein levels. Which way is it? Like, is it do we have more RNA than we would expect to see protein on, or the other way around?
So well, I don't think I could answer that in a general question, in a general way. What's interesting, again, all of these isotypes at the protein level, they're highly homologous, like 95%, right? If you look at the codon level, right, at the nucleotides, they use different sequences to get the same amino acid, and there's much less agreement at the nucleotide level. These are all separate genes, right? These are not alternative splices or anything else like that. So it's yeah. Sorry, it's unsatisfactory. I'll just, if I don't, I'll just tell you I don't know. Look forward to finding the answer. Yeah. Okay, we're moving on to kind of the second part, looking at the C-terminal tails. This is another region where all of the isotypes show a lot of variation. These tails in some branches of motility are called E-hooks because they have all of these glutamates, so they bear a lot of the charge. They actually have 40% of the overall charge of tubulin is in these tails. So electrostatic interactions are very important. This is also where a lot of these PTMs happen, the polyglycylation, polyglutamylation, detyrosynation, etc. Right? You see in all of these alphas that almost all of them have these terminal tyrosines, except we see that here we've got a phenylalanine and then TUB4A, which doesn't have one. There's not really any sequence conservation, but interestingly, there's rough conservation of length and rough conservation of charge. And that tells you that there's probably, the factors that are putting pressure on these sequences are probably more related to kind of their function and interactions. So going back to MD, right now, 20 years later, we have much bigger computers, right? So now we can run simulations here of a microtubule, and you can see all of these C-terminal tails here. I forget which is which, but one of them is colored in yellow and one of them is colored in kind of orange. So for the alphas and betas, okay. It was kind of fun to build these big microtubules. It's not a simple simulation to do, but the nice thing is we actually get a lot of dimer level data because I'm showing you a periodic image because we can build this in a box where it looks infinitely periodic. But if you look at, for example, these guys here, this is an image, right? So our entire system is actually 3 rows of dimers. So say 1, 2, 3, and then the rest are periodic images. But out of those 3 rows, we have 39 dimers, which means with our 500 nanoseconds, we're getting almost 20 microseconds of data out.
Just out of curiosity, the jittering here is just a Brownian motion of this?
No, this is all atom MD. I've just taken out the water and the ions.
I don't know what MD is.
So the molecular dynamics I described before,
yeah.
right? So this is the same protocol. So I'm looking at every atom, they're obeying these rules of the force fields, for example, right? We've got ions. We've set the ionic strength at 50 millimolar in these, temperature of 300, pressure of 1 atmosphere, etc. Yeah, go along there. Now, our experimental colleagues discovered that these tails have differential accessibility, right? In using the protein that actually does the detyrosynation, which is known as VASH, they found that it could detyrosynate the plus end where we have GTP tubulin, right, at this kind of growing end, but it could not detyrosynate tails that were back in this GTP state. So we go from kind of this accessible state to a less accessible state, or maybe even an inaccessible state in some level. Yeah.
So the enzymes that are known to do these PTMs to tubulin, are they specific to tubulin or are they acting on multiple?
So there's a lot of them that also function, for example, on histones and stuff like that. There's a little bit of crosstalk, not all of them, but there is an interesting kind of correspondence between the two.
So if you look at the regulation of those proteins, it's not going to give you much insight into what the post-translational and PTMs are going to do for the tubulin in general.
No, I would say not, unfortunately. Some of these PTM modifications, like the polyglutamylation and the I think that, as far as I know, I think that is unique to tubulin. I don't think that happens anywhere else. All right, so we want to investigate this now and see, how does these, what I often call these C-terminal tails or the CTTs, how does their accessibility depend on the state of the lattice? Right, so we perform these simulations, again, this is our analysis, and we're looking at where this tail interacts, right? It's a little bit washed out here, but you can see that this tail, with all of these red acidic groups, that's the negative charge, interact with a lot of basic residues around on different parts of the protein. And there's one site up here, but then you're seeing kind of these 3 other sites that we've colored either green or cyan or purple up in the top there. We find that once these tails find their binding site, they tend to stay bound there for a long time, right? Each of these binding sites has, apart from the first one, has multiple basic residues that they can interact with. Because of the multiplicity of binding, right, there's a lot of different conformations that could form, and that actually gives you an entropic benefit. Normally, if you would have something an unstructured peptide that would bind to a protein, you would think, oh, that would be strongly bound, but you pay a huge entropic penalty because you're restricting all of the degrees of freedom out of it. In this case, it can bind, so it's not the typical bound, but it's interacting with this site, but it's still got a lot of degrees of freedom within that region. Okay. So we do this analysis, we keep track of where it's making all these interactions. Then basically we calculate, this is what's called the Jacquard index. It's like a correlation, but it only counts when you're kind of in an on state. So if you imagine you sign 0 when you're not interacting with the residue and 1 when you are, if you just look at the ones, the Jacquard index you can consider to be basically it's the intersection, okay, for all of these groups. When I'm bound to 174, am I also bound to 380 over the union, if you will? Okay, so this is a nice way you can do that and then cluster, and then we see all of these sites which kind of show mutual levels of interactions without any kind of correlation between them. Okay, when we look at that, and now we do this same simulation in the GTP state of the microtubule, what we see is that the GTP, they start basically at the same conformation. The GTP basically stays where only about 10% of the tails seem to interact with the microtubule, whereas the GDP, and it's still running and growing, but it's increasingly, once it finds these binding sites, it stays bound, but it takes them a certain amount of time kinetically to find them. But then they tend to form these long-term interactions. If we look at these 4 sites that we talked about, so we see that the tail comes off, right off, this is helix 11, the tail comes right off of there, interacts very strongly with this group of residues. These are the same ones actually that Antonina found in one of her structures, for those who know who Antonina is. But then we see that the difference between the GDP state and the GTP state is that state 2 kind of seems to disappear, right? And that's one of the key points where those interactions, it helps kind of stabilize it and feed it down into the structure. When we look at the GTP simulation, we see that the GTP monomer is more dynamic in terms of the beta subunit, and that 4 of the 5 residues of that second binding site in site 2 actually see some significant increases in dynamics that we think just doesn't allow it to make those types of bonds. Okay, just in the last little bit, we're going to talk about interactions of the tails with themselves. Okay. You may or may not know about active sites and enzymes, but often there are acidic groups that sit in active sites of enzymes. And because of their close proximity, right, these are both negatively charged. So there's a lot of electrostatic repulsion between them. And they actually find it energetically favorable for one of them or both, effectively both of them to protonate and then hydrogen bond to each other so that they share this proton between them. Okay, so this neutralizes one of the groups and basically lowers the overall free energy. The way this works now in an enzyme site is that I've got these groups where they're protonated and hydrogen bonded. My substrate comes in, it blocks some of that electrostatic repulsion, but now I've got stored chemical potential energy in that proton, and it can do work to basically modify the substrate that's come into the active site. So the tubulin tails are the same thing, right? I've got all of these different acidic groups that, again, are maybe not fixed in close proximity, but certainly are next to each other and encounter each other quite a bit. And what we actually see is that they will come close together. For example, this is this glutamate here as well as glutamate here. They come together and hydrogen bond and form these kind of long-lived interactions. If we look at that procedure kind of in sequence, if you will, so here is this one E441 going to E446. You see that these side chains kind of approach each other. Eventually, you can get a proton, if you can see that there, kind of comes on to 446. It keeps getting closer, and then finally, after a few more picoseconds, it gets close enough where it forms this hydrogen bond. Okay. what's kind of interesting, and we're starting to gain insight on, is that these patches of glutamates actually help amplify this pKa shift. So the reason this changes is because of pKa, right? Where I've got these two charge groups next to each other. The pKa of one of them will shift from its normal value of 4.3 up into the 5.5 to 6 range, which allows it to protonate and then form this hydrogen bond. And having a patch of glutamates increases that electrostatic negative region, so it has even a stronger pKa effect. Now, in this case, interactions are not uniform. Okay, so this is a little bit of a heat map here showing the level of these interactions. This is the one that you saw before, where this is 441 binding to 446. But we see preferential interactions in regions that are like I to I plus 4, which kind of runs up this diagonal here. Okay. And when we look at the distribution of interactions, we see that sometimes we see a lot of interactions that are in the region of I to I plus 4, fewer to I plus 5, and certainly for the ones that are too close together. If you're too close together, there's too much basically mechanical strain and kind of bringing these side chains close by. If you're too far apart, the entropic penalty again kills you and you can't effectively form a connection, right? So this seems to be a little bit of the sweet spot, but then you see even for that range of interactions, we see a pretty wide range of radius of gyration. So they can form relatively compact structures with an average radius of gyration around 4 to 5, but then also more extended structures around 10 to 12.
Can you, there could be hidden Markov models within this. So can you develop like some sort of models to understand the kinetics of these interactions?
Yeah, I think we could, because we've got enough data. So we're running, I didn't go over the details, but these are what are called constant pH MD simulations.
Mm-hmm.
So most of the normal, if you will, quote unquote, simulations we run, we fix the pH, right? And which really only means looking at histidines and figuring out whether they're protonated or not. But in constant pH, basically we do Monte Carlo. We run for, and we just basically keep saying, do I want to add a proton onto this group or not? And so based on its proximity and where it happens to be, it may protonate that group. So it's a what we call a grand canonical ensemble, because the number of atoms is changing then, right? I take a water molecule and I'm adding a hydrogen here, so I have to introduce a hydroxyl somewhere else to kind of balance the charge and stuff like that in the system. So I think we've got enough data in these simulations that we could probably do that. If you remember what those patterns look like on the last slide, and unfortunately, all I'm doing is just showing you the data here, right? When we look at tub B2, tub B or tub B3, we see different patterns of interaction. In tub BB, we see strong interactions in the N-terminal part, slightly weaker interactions up in the C-terminal part of the peptide. Tub BB shows interactions kind of in the middle, if you will, towards the N, and very little at the N-terminus. And then tub B3 is just, again, different and balanced all again. I have no part of the story to tell about what this actually means, right? It seems likely that these different isotypes, because their chains will have different levels of interaction, will have different interactions with microtubule-associated proteins that would come in, but that part of the story has not been completed yet. So that's work to come. If you invite me back in 30 years, maybe I'll still be alive and tell you what it is. All right, so quick summary then. You've learned more about tubulin isotypes than you thought you ever would. They have subtle sequence differences in that they produce not so subtle changes within the protein. These effects appear to include protofilament shear and the ability of the interactions between protofilaments, and then how that might actually affect twist of the lattice, and then how MAPs would bind to the microtubule, certainly through these changes in the C-termini. I'll point out again, this is such a very small piece of the tubulin code landscape. We've only looked at a few isotypes in a few isolated situations, but we're starting to see some interesting patterns, which hopefully we can relate to experiments in the near future. And with that, I will finish First of all, highlighting some of the people in my lab, Arpita, one of my early students who showed some of her first simulations, a postdoc I had, and research scientist Dr. Hoon Sim, and a current student, Patrick Delir, who's really doing a lot of the work. My collaborators at U of M, Kristen Verhey, Lauren McCormick, Mike Scanfrocco, and Morgan DeSantis, who is a great collective of microtubule labs. And then my other collaborators, Gary, first and foremost, because his name starts with B, is at the top of the list. So there we go. And Will Hancock, Lauren, Jeff, and Dan. And I thank you for your attention.
Questions?
Yeah, so the Tubulin code, what you showed us is it's more of like a functional code than like an information-based code, like DNA, like if you look at DNA, like the structure of each nucleotide doesn't really matter. It's the order it's in that's important.
Yeah.
But in your case, it seems like for the isotypes and all the PTMs, they all seem to have actual functions. Do you agree with that?
I agree with that.
So
Yeah.
code may be not an appropriate term or something?
Oh, wow. Okay, if you want to bicker about
Yeah, no.
So yeah, so I would argue that it still is, it's a code, but would you prefer signal? Would you prefer something else? It's an indication, I think, of what function could be. Like I think, even like I've shown some differences, right? I have no clue, for example, even in these lateral bindings, you see all these differences in terms of hydrogen bonding states or secondary structure and salt bridge states, for example. I have no clue functionally what that does. I have a feeling that it's changing mechanics. We have some sense that it's changing the preferred angle of protofilaments, like when they bind, how much the rotation up a bit in there. But functionally, we're a long ways off from that. So I'll store that, and if we agree the code is not the right word, well, it's not mine, hard to decide. But
No, that's good.
yeah.
Any other questions? Go ahead.
No, this is also kind of related to the tubulin, the different presence of the isoforms. Another question about that, but you said that monomers are very unstable. But so the kind of ones that survive will be the ones that form dimers kind of quickly after they're produced. Is there preferential binding between different tubulin isoforms, which would mean that there's certain interactions that are going to be more prevalent, whereas others that are going to be sort of unhappy?
The simple answer is we don't know. I'd be so if you look at sequence conservation, in between the dimers at that interface between alpha and beta, that's almost 100% conserved within dimer, and it looks different than, even though alpha and beta are highly homologous, that's different than the interface in the interdimer interface along the protofilament. So it's that interaction is stronger within a dimer, and it's more highly conserved. But yeah, we just don't know. That's a great answer for everything.
I was wondering about the scale of the last part of the data that you were showing us, where tails would interact with themselves.
Yeah.
And I'm wondering if you think that it's like, is it a huge change where it's like, one tail will fold a little bit and one tail folds in half, and then a microtubule associated protein would recognize the difference in that? Or is it a much more subtle change?
So it tends to be, like I said, it tends to be like I to I+4 types of interactions. So it does tend to form a loop in what we predominantly see.
Okay.
What's kind of interesting with that example I showed where 441 binds to 446 is that it binds that loop, and then 445, I think, is one of the glutamates that gets preferentially polyglutamylated. Mm-hmm. So it's almost like it's maybe presenting an epitope that could be recognized by an enzyme coming in to modify the tail. Hypothesis, but I think, and these interactions, it's still pretty dynamic too. We expected to see in our simulations that once one of these forms, then maybe that would put other parts of the peptide in close proximity, that they would form another salt bridge or hydrogen bond, you know, and then et cetera. We've only seen 2 to 3 bonds form a handful of times, and it's not. We thought it might actually form a very stable structure, but we don't see that yet. So, yeah, it's kind of interesting.
Any other question?
Hey, Ryan, sorry. I was wondering, your results on the nucleotide state and how that would affect the tail were really interesting. And, there were sets of maps that seem to put tubulin in a compacted state and sets of maps that seem to put it in an expanded state.
Yeah.
Might act like nucleotide state, I'm not sure. Like, what would you predict in terms of like the compaction state to the accessibility of the sea turtle?
Yeah, so you're pushing me to betray one of my strong biases in that. I think when you look at these parameters, when you look at the offset along the protofilament, there's very clearly a compacted and expanded state that's dependent on nucleotide. I think we kind of go a little bit astray when we start assigning that to other things that we see when we have kinesin decorated microtubules or Taxol decorated, et cetera. Because there, I think it's more the twist that's actually being the signal rather than the compaction or expansion.
The twist along our protofilament?
The twist between around the microtubule, right? That's where you see this little bit of a shear that actually gives rise to this kind of superhelicity. I think that's where the differences come. And it's not, we tend to still think about it in binary, saying, oh, look, double Corton is, or does it expand? Compacts, okay, expand. But I think we're biased by the structure that we have because we're always looking at microtubules that are fully decorated with X, Y, or Z. If, to see a structure with a single kinesin bound, it'd be really interesting to see what does the lattice do around that structure, right? Around that piece, or what if you only had 30% Taxol binding along the microtubule, right? What would that look like? I think there's a lot to figure out, and we're kind of biased right now in what we see. Yeah.
Right.
So like, do we see the post translational modification uniformly along the microtubule molecule, like the filament, or is it only at the ends where polymerization?
That's another good question. So, we do see some correlations between different PTMs. It's almost assuredly not uniform. Like you'll see, there's different, and it also depends on which cells you're in, for example, right? In that, in axons, you see really high levels of acetylation, for example, and acetylation, I think, is correlated with deterioration. Is that right? I was looking at my colleagues to give me help.
Yep.
It's like, so we don't know enough.
I mean, I think we're limited by antibodies. Like the only way we can detect these things are with antibodies. The antibody's like 60 nanometers large.
Oh.
Okay, so the antibody that is recognizing this tiny epitope, but it's this big, huge thing. So if you stain cells, yeah, you see like a lot of uniform antibody staining, but that could be hiding significant amounts of variability that's happening on smaller spatial scales and are currently detectable by the antibodies. There's been a big push to have live cell probes or peptide, other types of proteins as probes, and there's some traction there. Actually, Justin can talk about some of the probes that he's working with. But until we have, I would love to have like a small molecule probe that binds to a tail in a specific state. Then you might actually be able to do proper super resolution microscopy or something that would get you to the spatial scale that would answer your question.
Okay, great.
But
I was
Yeah, go ahead.
because I was thinking, like you mentioned, I think one of your results is that changing from the GTP to GDP actually kind of hides the C terminal.
Yeah.
I saw in like basic biology class, it's like the GTP to GDP determines it's like polymer. Like it makes the prompt to disassembly.
That's great.
So I was thinking, do you think C terminal could play a role in like controlling, like maybe signal where disassembly or I should assemble one
Great question.
function?
Yeah, like, I know, like, so the C-terminal tail does play a role in polymerization at some level, in that the tail of alpha can actually come around and interact with the bottom of the diamond. And that actually, if you cleave it off, you actually see that it polymerizes faster than if it's there. Functionally, I like, it's a cool result, right? And we've seen these in simulations for 15 years, right? Running, as soon as we put the tail on, but functionally, I don't know if there's a role there for regulatory as well. I don't, not that I'm aware.
Any other question? Can I ask one more, last
can,
one?
sure.
So this is a person from outside the field, and I'm gonna embarrass myself asking that question. So here you're talking about tubulin. How generalizable some of these concepts to other proteins? Like, I mean, is there like
Yeah.
every single protein, do you have to do analysis and there will be new set of interactions and?
Yeah, so I think we've been we've been so strongly schooled in sequence, structure, function, and people who study phosphorylation, think, oh, this is, managing all this regulation. I think as mass spec is getting better and higher resolution, being able to learn, we're learning, like, there's just so many modifications that we're seeing, post-translational modifications coming out. I saw a talk from this woman from Iowa who studies nuclear actin, right? When they isolated it and looked at PTMs, this is a protein of 375 amino acids. It had 140 PTMs.
Oh.
So is that what is that? If it's half PTMs, right? What's it doing? And I think there's a lot we don't know. If that's not clear from every other answer I've given.
Okay, the chat, do you have any questions? Yeah, I guess not. So let's thank our speaker on my side.